the cytoplasmic organelles. And we have here particles are, what are these particles present in the cytosol? It could be proteins, mga lipids, other organic carbohydrates, nucleic acids, okay? And they may also contain organelles with distinct structure and function. We'll be discussing them. And some are said to be visible under the microscope. Okay, now let's talk about endoplasmic reticulum. Now, endoplasmic reticulum is found in a very near proximity to the nucleus. Now, endoplasmic reticulum, or we call it as ER, can be of two types. You have the rough. Now, what makes this rough is because of these ribosomes, which are attached. And we also have the smooth endoplasmic reticulum. Now, for the smooth endoplasmic or smooth ER, it does not contain attached ribosomes. What is now the function of the smooth endoplasmic reticulum? Now, the smooth ER is the site for lipid synthesis. It's the site for lipid synthesis. So, the lipid molecules are now synthesized here in the smooth ER. Now, for the rough ER, this is the site for secretory protein synthesis. So, all those secretory proteins are synthesized in the rough ER. So, these are ribosomes, the picture of the ribosomes. If you remove this, kanimurag bugas-bugas. Now, you could see here a two subunit structure. Dua ka subunits na siya, the larger and the smaller subunit. Those are ribosomes. Ribosomes are sites for protein synthesis. So, some are attached to the ER, so you have the rough ER, and this one doesn't have attached ribosome. Now, Golgi body or Golgi apparatus lies in a very close proximity with the endoplasmic reticulum, especially the rough endoplasmic reticulum. What's the function? What's the function of this Golgi? Now, the function of this Golgi is for packaging and secretion. This is where the immature secretory proteins from the smooth ER is directed for maturation, or for packaging. So it's made up of transport vesicles. So this contains also membrane. ER contains single membrane, and the Golgi as well consists of membrane, a single membrane structure. So it has lumen. So it has the cis phase. Now the cis phase receives immature proteins from the rough endoplasmic reticulum. And the trans phase receives matured proteins which are about to be packed or sorted, which will be distributed to the different parts of the cell, or maybe to different parts of the body. Okay? So that's the role of the Golgi. That's how it works. So from the rough ER, this is now the direction of the protein or the secretory proteins. So from the rough ER, it is transported to the cis phase of the Golgi, then to the mid phase, then to the cis phase. Then later on, this portion, that one, will be pinched off, and that becomes transport vesicles. So, what is inside? That contains the matured proteins. So, that will be destined for what? So, these proteins can be destined for exocytosis, or say maybe, needed for secretion. So, it will be for secretion. Or maybe part of the cell membrane. Okay? That's how it works. Okay, next we have lysosomes. Now, This is for intracellular digestion. Now, you have here the lysosome contains hydrolytic enzymes. This is hydrolysis. Now, those unwanted molecules or unwanted organelles, no? Those old organelles, like for example, an old mitochondrion or mitochondria, they will be engulfed by the lysosome, and that will be digested by the hydrolytic enzymes or digestive enzymes, and that will be destined for recycling. So, you know, cell go for recycling. So those unwanted molecules can be engulfed or swallowed up by these lysosomes, and that will be digested, and later on that will be released for recycling. So, are you doing recycling in your homes? Or maybe in your dormitories? No, it's good for the cells. It's practicing recycling. Okay? So, this function help in digesting large molecules. Or there are also some foreign microorganisms like bacteria and viruses that would be engulfed by these lysosomes and digested by its hydrolytic enzymes. So that will be degraded. And of course, you have here, for those worn out organelles like mitochondria I had mentioned a while ago, now they will not be thrown out. They have to be engulfed by lysosomes, and that will be degraded by the digestive enzymes and ready for recycling. Now, another function. Now, by the way, in the plasmid reticulum, Golgi, lysosomes, now they are present in eukaryotic, absent in prokaryotic or bacteria. Now, except for plants, lysosomes don't have lysosomes. Animal cells have. Okay. Now, another we have vacuoles. Now, vacuoles are present in both plants and animals. However, plant vacuoles are too large. They are larger, much larger than animal vacuoles. What are they for? They are for storage. Storage or store various substances including waste products. So mga waste, ipangdanhok nila na diha. They are dumped in the vacuoles. And that also maintains osmotic pressure, store food particles for amoeba. You are familiar with amoeba. Amoeba histolytica, the one that causes amoebiasis. So that vacuole is used for their food storage. And that also provides turgidity and rigidity to plant cells. So what makes plant cells rigid is also because of the vacuoles that contributes as well the rigidity. Another, we have the mitochondria. Mitochondria, this is made up of two membranes. You have the outer membrane and the inner membrane. Now, like the nucleus, mitochondria or mitochondrion singular consists of DNA. Now, what is this DNA for? This DNA is for maternal inheritance, maternal traits. Meaning, these maternal traits can be passed on from mother to offspring. The father cannot contribute. The father cannot contribute. Only the mother. So, because what gets inside the ovum of the egg cell is only the head of the sperm. And the mitochondria are found in the tail of the sperm. And that's why only the nuclear DNAs which found in the head of the sperm fuses with the ovum or the egg. So the mitochondrial DNA will be degraded together with the rest of the tail parts. So the father cannot contribute this mitochondrial DNA, only the mother. So it's made up of outer and inner membrane, and the inner membrane forms a continuation. We call that as the cristae. These folds here, the inner folds, we call that as the cristae. And you have this space here, we call that as the matrix. Now, you have here the green called the ribosomes. So it contains ribosomes, it contains DNA. That's why, according to some evolutionists, long time ago, this mitochondrion was bacterium. It's actually a cell before, according to evolutionists. And there was engulfing without digestion. So, engulf niya siya, that's accordingly, and that contributes complexity. No? But, of course, we don't believe on that. But that is according to the evolutionists. Because of the presence of the DNA in the mitochondria and, of course, the ribosomes. Kung nasa yung DNA, pwede siya makasynthesize of proteins. Because nato siya ribosomes. Proteins are sites for ribosomes are sites for protein synthesis. And the one that regulates is the DNA. No? Okay. So, what is this mitochondrion? This is the powerhouse of the cell. That synthesizes energy-rich compound ATP. So, it's the one that's the site for the breakdown of your food to become ATP or energy. Now there you have plastids. Now plastids are double membrane organelles like mitochondrion. They are responsible for food making or photosynthesis. Okay? So plastids can be of three types. You have the chromoplast, the chloroplast, and the leucoplasts. And the leucoplasts, okay, of course you have the chloroplast, which contains the green pigments called the chlorophyll. Leucoplast doesn't have any pigment at all. No? So puti siya. You have the chromoplast, it contains now the red or the orange pigments. Leucoplast contains this amyloplast, brachioplast, ilioplasts. Okay. So, what makes these lovely colors is because of the chromoplasts. The carotene, the lycopene, okay, xanthophyll, they are found in the chromoplasts. Okay. Leucoplasts, colorless. Like for example, starch, mga greens, then oil. So, they are found in the storage organs like fruits, tubers, and seeds. Now, for the chloroplast, you have here, it's a double membrane structure, the outer membrane and the inner membrane. Now, here, unlike the mitochondrion, the inner membrane is not, there is no continuation here. For this, you have here this structure called this peso-like structure or the coin-like structure called the thylakoids. The thylakoids. And we have the grana. This one, granas, grana, plural, granum, singular. chloroplast stroma. Then we have these stylacoids, or the coin-like structures here. And this granum is made up of one pile of these stylacoids. Function, this is for the site for photosynthesis. Then we have the centrosome. The centrosome consists of pair of centrioles. You have here, pares yun na siya permi. Pair, pares. Which of course made up of microtubules. Function, these help in the movement of chromosomes during cell division and in the formation of the hair-like structures called cilia and the whip-like structure called the flagella or flagellum. Okay. Another, we have this. Okay, we have here cytoskeleton. Cytoskeleton is made up of microtubules and microfilaments and mga intermediate filaments. Cyto, you have the word cyto means cell, skeleton. This gives what? Shape of the cell. So kung walay cytoskeleton, walay shape ang cell. So what contributes to the shape? It's because of the framework of the cell. We call that the cytoskeleton. Could be microtubules, microfilaments, and mga intermediate filaments. That gives also structural strength to the cell and responsible for cellular movements. You know what cell moves? Organelles move. Chromosomes move. And what contributes to their mobility? It's because of this cytoskeleton. Okay, we have these differences, a prokaryotic and eukaryotic cell. Nucleus is underdeveloped. Here in eukaryotes, they are well developed. Now, prokaryotes or bacteria, only one chromosome is present. Here, more than one. Then membrane-bound organelles are absent. Like for example, Golgi, katong na yung mga membranes nga organelles. This one, present. And the size is 0.5 to 5 micrometer. So mas gagmay sila compared to eukaryotic cell. So they have that examples. Bacteria and the blue-green algae and the rest organisms belong to eukaryotes. We have differences of animal and plant cell. Animal cells, generally they are small and plants are large. Absent cell wall. Cell wall is present here. Plastids are absent, present. Vacuoles are smaller, here larger. And centrioles present. Here in plant cells, they don't have centrioles. Itong centrosome, centrioles. Usually, these are very important in the cell division. Okay, so that ends our topic. You know what? As you look at how you are made of, can you see a designer in there? Can you still say that you are a product of chance? Now, when we look at the cell, now we could see God as the designer. All the organelles are highly organized. And that just shows that the God who designs us